So I think I get an advantage here today because I can work in the back and try to make my slides while everybody else is speaking, and be able to mix in some things from the talks today that kind of are relevant at the same time. So there'll be some wording changes that are reviews of some of the things you've heard today so far, and a little bit about where we're at and maybe where we really need to be, because although we're making a lot of progress in many different things right now, we have to ask ourselves where that progress is being limited right now. And as Matt said, I think a lot of that is related to phenotyping. There's several breeds right now that are producing genomically enhanced EBDs. I think I might even have one more breed than Matt had on here. I think I found 10. I'm not sure which one's different yet, but we can figure that out later. We've been approached, and by we, I mean those of us at Clay Center, and I'm sure people, several of the other folks in the room have been approached by several other breeds in terms of trying to get this going. In fact, I can think of 5 other breeds right now off the top of my head that are saying, hey, we want to get this going in the next year, the next 2 years or something like that. What do we need to do next? So it's happening all over, and the reason why it's happening is because things are working pretty well. There's been a tremendous amount of progress. We go from a paradigm though right now where traditionally the picture on the left is our black box process, where my black box is green in this case, where we basically take pedigree and performance data, go through a traditional BLUP system with a relationship matrix calculated using pedigree information. And now we take that same 2 sources of information and mix in a little bit of high density genotyping at the same time and go through a new green box and get a genomically enhanced EPD through one of several different methods, some of which Matt spoke about earlier, with blending, with single step types of approaches, with maybe using correlated trait approaches, whatever it may be, but this is what's going on right now. And what I want you to notice about this, and I'll keep harping on this through this whole talk right now, is that the 2 boxes on the left at the top are pretty much exactly the same here in the traditional method as they are in the new method. Those boxes do not change. There's no new information right there. And that's a bit of a problem. I'll tell you why here in a second. So a lot has been accomplished so far. Breeds are taking real advantage of this technology right now. There are breeds that have way more genotype information, way more animals genotyped than there are markers on the 50K SNP chip anyway. And there's been a tremendous benefit in terms of genetic gain achieved right now and being achieved in the future based on this. But right now, data limitation is still our main hindrance, and that's particularly for the case of novel traits. So what do I mean by novel traits? Well, we've got 2 different categories of trait complexes here right now, right? And this goes back to those 2 blue boxes being the same at the beginning. We've got the traits that are measured on National Cattle Evaluation as a standard right now. This is the typical data that your seed stock breeders are turning in as a standard. This is what it takes to get their cattle registered. This is what they've been collecting to get new traits developed within the National Cattle Evaluation, those sorts of things. And then we've got everything else. And the everything else is where the rubber hits the road for many of these genomic information talks. This is what I'm going to be terming as novel traits from here on out. These are the traits that are not well measured in National Cattle Evaluation, be it because it's a smaller breed and they only measure growth traits right now and have a hard time getting that accomplished, or because it's a larger breed and some of these things just don't measure very well. They don't come to us very well in our standard databases that we're able to collect, because it takes a real amount of labor, time, and expense in order to get these types of traits recorded, okay? You guys think about them in your head. I'll have a list here in a second. I think the three talks we've had before really describe this. And then the other thing I won't cover much here, but we have the additional complication of almost everything we've talked about today is in the context of working well within a population, or for our cases, for our purposes, you might say within a breed, and maybe don't work as well when we're talking about taking those predictions across breeds. So how do we start to have a real impact in development of novel genomic predictions? So I would go back to probably about 8 years ago, and the main reason that seed stock breeders and breed associations were approaching those of us in the research community on why they needed genomics was because they were interested in these traits that are not being measured very well right now in national cattle evaluation. Okay, now granted, we're able to make a lot of impact on some traits that are difficult, but in national cattle evaluation, like heifer pregnancy, for instance, might be something that's measured, especially in whole herd reporting context. We might have some maternal milk that usually would be measured late in life that now we're getting a much earlier prediction on things like maternal milk. There's several examples like that, but the things that people were approaching us wanting were things like feed intake, feed efficiency, that we mentioned earlier, disease resistance and fertility, and maybe going outside of that a little bit more to some longevity traits, food safety, survival, color stability, meat quality, meat yield, some of the things that we don't do well or we only do by proxy of some of the things that we can measure, for instance, with ultrasound right now. The difficulty in measuring these things translates to a difficulty even in a research setting of trying to do these things well, because it takes a lot of time, money, labor, whatever you want to say. And this is really the fundamental problem with the genomic development in these sorts of traits complexes, because we can't accumulate the data that we need to do this well, okay? So I'm going to talk a little bit. We've covered this earlier, and I put this slide in there because we hinted at it a bit earlier. If we talk about the economically relevant traits that Dare sort of talked about a little bit earlier today, there's several examples of things that affect the whole beef industry here. And I know in context of a commercial cow-calf producer, you might not have all these be economically important to you as a cow-calf producer if you're selling at weaning weight. But sometimes there's some of these things that affect the feedlot and affect the packer that if you want to sell next year, you may have to select for in your commercial cow-calf operation too, or they might not get bought by the same person over again. Okay, there are a lot of these things that are measured in the feedlot sector, measured in the harvest facility, whatever you want to say, that are not being done well right now in our current national evaluation system, and obviously some of these are. What can we do to address these a little bit better? Okay, and some prime examples, basically everything that I've highlighted here right now are things that in general we have a proxy measure in our national cattle evaluation in yellow, or we really don't have much of a measure in most of our genetic evaluation systems at all. Or if we do, it's on some small subsets. Okay. So the traditional way that really we've gone about several of these traits, and this would be the traits that I highlighted in yellow earlier, have been through the use of indicator traits to try to do a better job of adding accuracy to these trait complexes without actually measuring the actual trait. You guys, we talked about birth weight for calving difficulty earlier and had some discussions with Arlen about that a little bit earlier. We can keep talking about that later. We have a ribeye area for yield that we use as a measure of overall yield. Is that really the whole picture for yield, Mike? I would say not necessarily. Scrotal circumference for male and female fertility, ultrasound in breeding animals for carcass traits, treatment and lesions for disease incidents, but none of these things are exactly the trait that we're looking for. So really, I would propose that genotyping is our next level of indicator trait, okay? Really, we don't care what the genotypes are inherently on our own. We care about how they affect the things that we're interested in. And genotyping is just the ultimate indicator that we use to add accuracy to our overall national cattle evaluation. And hopefully we can extend that then to these novel traits, okay? So in genomic training, we need to estimate the effects of the genotypes on our traits of interest. And obviously that can be done several different ways. It can be done on a marker by marker basis, or it can be done using methods that incorporate all of the markers together into an overall genomic relationship matrix in some fashion. The problem is that we need a large phenotypic database in order to do that well. And then the other big problem with that, this relates to those populations earlier, and I would argue that this could even affect within breed predictions, is that the inference population that you do a really good job with are the animals that are actually phenotyped. And it falls away the further you get away in terms of relationships from those phenotyped animals. Okay? How can we do a better job with that? Correlations then are reduced with small data sets or weak relationships to these inference populations. Really the problem is we're trying to predict effects for 50,000 or more markers, usually more nowadays, and we're trying to do that with a whole lot less data than 50,000. And so how do I say that this is the effect of 50,000 markers when I don't even have 50,000 points? I can do it perfectly or I can do it not at all, basically. And obviously methods take those sorts of things into account, but they're hardly perfect. And that's really what the inherent problem is. I think Steve said earlier this morning that there's many analysis methods where the simplest method is actually the best. This is part of why. Because sometimes doing something fairly simple when we have this low power right now is something that is actually not trying to estimate too much and just trying to help you go in the right direction. Make a simplistic assumption and move forward. Okay. So this is our situation. I think for most of these novel traits, I think we're in a desert for phenotypes right now. And even though we've had these large efforts that we'll talk about here in a minute for trying to increase our phenotypes right now, there's about as many phenotypes for affecting these traits that we're really interested in as there is water in this picture, okay, as a percentage. It's just not there. And overall, there's just a real lack for this data, and even more so if you take things like fertility that we're measuring, trying to do with whole herd reporting in our seed stock operations, and say that's the same trait as what we see in terms of fertility or longevity in our commercial data sets for our cow-calf guys, okay? So what are strategies and what do we need to be doing? And that's pretty much where I want to close the talk. I'd like, you know, I know we have some time for discussion after that. I don't think I'm going to be complete here at all. This is where I think we have to think as a body of progressive commercial and seed stock producers and a body of breed association representatives and a body of research scientists. How do we progress and try to do a better job with this? I would argue that the two places that we've been focusing on or need to focus more on is trying to increase our research institution data collection. I'm partial to that working here at the Meadowlands Research Center, so I can't leave that one alone completely. And then trying to increase our commercial partnerships to make sure more of the data that's affecting the commercial entities that we're trying to target with these economically relevant traits useful for commercial producers are being brought into the system so that we can do something with them, okay? It sounds easy, and I realize it's not. Let's talk about some of the issues with that. So just research institutions, I don't need to spend much time on this. We just heard about 3 big USDA NIFA grants that were related to BRD resistance, feed efficiency, and fertility. And then there's several other grants going on and further in BRD, liver abscesses, vaccine response, immune function, E. coli. These are just a few that I know about. That doesn't cover everything at all, I'm sure. There's several grants going on right now. And here at the Meadowlands Research Center, we collect data on about 3,000 plus animals born every year in the germplasm evaluation program. How much does that really contribute to the whole data set overall? But that's what comes from this data source right now. This is the trait complexes that we evaluate here. I would say that although I said there were 3,000 records, there's none of these that are actually recorded on all 3,000 of those animals every year. Very difficult to get scrotal circumference on the heifers, those sorts of things. So it just doesn't happen. We do the best we can, and this is just an example of a lot of traits we try to record here because we feel that there's a real value in recording as much as possible. Okay, so even though all this data is out there at the research institutions, and most of these efforts have resulted in thousands of phenotypes for novel traits, in some cases more than 10,000 records. We were at, what, 8,500 almost, Bob, on feed efficiency in the grant. we'd be a comparable number just here alone, so that's over 10,000 together. We're still much lower than the amount of data that's in the NCE databases, where we're getting these higher correlations, but not perfect correlations of the 50K chip to predicting our actual genomic performance. And you know, there are single sires in several breed associations that have more progeny from one sire than what we've been able to collect in terms of our novel traits. Just think about that for a second. Our 0.99 accuracy sires that we have in some of our breeds, some of them have, what, 65, 70,000 records, progeny records? We're nowhere near that on novel traits. And that's the data, that high accuracy sire, that's the data we're training with on the NCE traits. We're nothing like that on our novel traits, okay? Not even close. We're further complicated by the relationship of our research data that we have right now to the overall industry for two different reasons. One, how well do we tie to the important pedigrees in the industry if we're using marker chips that are kind of based on a relationship? How well are we tying, say, Angus used in a grant to the Angus of that covers the whole breed, right? How well do we do that? Or how well do we do it when we try with a very difficult effort actually to make sure that our sires at the Meat Animal Research Center are tied to the industry? Then we have all these crossbreds that we're trying to have this prediction work in several different places, okay? And that's difficult, all right? So alternative usage strategies become very important. What I mean by that is we need to develop different analyses to use these research data well, because using it with the base 50K chip methodology and beyond that could be a little tough from any research institution. I do think there's reason for optimism with some of what Megan was just talking about with functional chips. Whether this version of the functional chip is the one that does all this or not, I think it could help us try to get somewhere, and I'll talk about that in a second. Commercial collaborations. I've had discussions with several breed associations in terms of trying to collect more data. And I think there's been news releases, things like that, from several breed associations about trying to offer commercial cattle producers an incentive of some sort, cow-calf guys particularly, in order to have them collect more data, and when they use sires of those breeds or that breed that is involved, try to have them use more, collect more data, and then basically give them something back in return, be it an EPD or an overall prediction of mean progeny performance or something to that effect, a producing ability on the cows, or whatever that might be. Give them something back that incentivizes them actually recording that data, okay? Now, that can be tough to do, but let's think about that. That's the cow-calves. Feedlot and abattoirs might be tougher. I'll talk about that in a second. Potentially, what we've got here is millions of animals in the industry right now that if we were measuring 1% of, we could probably do better than a lot of our seed stock data that comes in every year. Okay, think about that. If we were able to actually try to get enough commercial entities involved to actually do something well, we could easily surpass our seed stock databases. Okay, it wouldn't take that much. It would take a lot of work and effort, don't get me wrong, but it wouldn't take that much of that data to actually surpass what we have here. Now, granted, phenotypes are often more or less precise when you're talking about this type of data, but you have to think about what you have and have to think about numbers quickly passing over the requirement for precision. Okay, so cow-calf operations. I think the capacity to record is the most challenging option here. When we talk about trying to do something, we need to talk about database types of tools that we can offer in as simplistic a form as possible to our commercial producers to allow them to actually record. And I don't think that's just an Excel sheet. I think it needs to be simpler than that, okay? Trying to take them from something and trying to use Brian's program, Allison, would be very difficult for some commercial producers. I think seed stock, I agree, is a good target for that. But trying to do something like that may be difficult, but there are baby steps maybe we can do in between to try to make sure that type of producer actually has a place to put in at least their cow inventory every year, okay? Just putting in their cow inventory every year and whether or not they wean the calf is a tremendous amount more data than what we had before. Some of these producers only see their calves twice a year. Some of them only see them once, and that's when they sell them. That's their inventory, okay? That's still useful because if we can tie that somehow with a tissue sample, potentially without a tissue sample, if we can at least get the dam and know where the dam came from, it still ties us back to something else. Linking to NCE databases can be a challenge with this, partly because it's difficult to actually know the pedigree, or it's difficult to actually get genomics and actually individually genotype every animal. I don't know that I want to pay for that either. We need to think about strategies. We'll talk about that a little bit here in a second. And then again, I think this sector is probably the place where we have the best chance of incentivizing our process in order to get the cow-calf producer to give us a little bit more data. I think here is where we can offer them something like predictions, marketing strategies that they can use to say that their product is more or less beneficial than their competitors. Feedlots and abattoirs, I've lumped these together right now as margin operators, and why I've lumped them together is because I think incentivizing each of these entities is going to be a lot more difficult. We've spoken to some of these before, and I've spoken to them with some of you in the room before, and they're definitely are generally not interested in paying a premium for calves, even if the calves are better, even if you can show the calves are better. They're generally not interested in paying a premium. Whether we can convince them they're wrong or not is arguable, but they're usually have a better chance of docking cattle they think are worse instead. That's the reality of the situation right now. Whether we can change that or not, I know there's programs out there trying to change that right now, but that's the situation that we're in. And getting them to actually give us data so that we can make some of these things better is a little bit more of a different proposition. I think it probably requires a labor and a cost, a monetary donation, if you will, in order to get that data from each of these places. It could be very valuable, both entities, very valuable, but generally if we actually want the IDs tied to the data very well, I know there's the IDs and I know it all works perfectly, but if we actually want to be sure that the IDs are tied to the data, if we're talking about harvest facilities, if we want to make sure that we're getting IDs tied to feedlot records, we probably need to make sure that we're putting some extra incentive into it on our end, because it's probably not going to come out just from a feel-good attitude about the industry. That said, this is where we can have a huge impact, okay? And I'll leave it there. I think we can do a lot with these, but we need to think about strategies to do it. How do we use this data from alternate sources? There's three things I can think of right offhand. I think the functional chip could help us a little bit, use our research data a little bit better by actually maybe having a better chance of being near causal variation than what we have been traditionally. Talk about that in a second. DNA pooling, I've talked about in several audiences before, but it may give us a way to use a lot of this data without actually individually genotyping each animal. And then where I said that commercial cattle must tie to seed stock, and this needs to be using either parentage to get pedigree ties or using genotyping to get genotyping relationships, maybe some of that is where DNA pooling can be useful. So I'll talk about those things really quick. Functional variants, I think Megan gave an excellent overview of what those functional variants are and what they mean. If you don't get the protein, chances are something's not going to work right, okay? Well, look at it in the context that's true with embryonic mortality. Maybe there's cases where something just doesn't work as well. And there might be cases where the protein still works, but maybe the growth hormone pathway doesn't work as well and we don't get quite as efficient of growth as we used to. Okay, something to that effect. Those are the types of things we need to think about for all of our traits with these functional chips approaches. I don't think the iteration that we're on right now is going to be our final iteration. I think we're going to have to keep doing this and keep looking with gene annotation, getting better at regulatory regions outside of the parts that Megan mentioned before. But we need to get a stab at this and see if getting closer to causal variation can actually help us add accuracy with a little bit lower power, okay? I won't go into this much, but just know that we're making an effort similar to the fertility grant of using that functional chip here. We're trying to make sure that we genotype founders of the germplasm evaluation program here, around 2,000 so far, so that we can impute those functional variants into our whole GPE pedigree. The weight traits project that all of you are here for right now has invested in genotyping of 2,000 animal, including around 500 that are part of the feed efficiency grant, okay, that several of you have been involved with. We also have a project with Gary Bennett, is in the back of the room right now, is in charge of, where we're selecting the former marker-assisted selection lines to reduce the number of loss of function alleles and see if that selection on those markers can actually improve some of our quantitative traits, including potentially fertility. So, okay, so DNA pooling. This is an approach that we've used in research demonstrations with a lot of commercial entities so far to try to find markers that are important for some of the traits that are more difficult to measure, some of the novel trait complexes. And basically the concept is you take a bunch of animals, say 100 or so, that have a really good phenotype and 100 animals that have basically the opposite phenotype, put them together. I don't have any molecular folks in here, so I can say you mix them up in a test pood by shaking up a whole lot and genotype it all, okay, right? And essentially you're getting gene frequency estimates between those two different pools. Those cases where one pool has a very, very different gene frequency than another pool, the other pool that's the opposite, are strongly indicative of markers that would be important for the trait that you're separating those pools for, okay? We've used this so far for fertility. We've used this so far for respiratory disease in terms of lung lesions. We've used it for liver abscesses, and we're seeing some pretty reasonable results, I think. We need to do, I'm not going to remember the other word for validation either, Matt, but we need to make sure that they work. Thank you. This is the problem when you have a word ingrained, even if it's a bad word. So anyway, we need to make sure that they work well in a separate population, but we're actually seeing some promising results somewhere. And we're seeing a tremendous cost savings relative to individual genotyping. In fact, a paper that we have in JAS a couple years ago used lung lesions at a slaughter plant, and all we used, we didn't do any contemporary group or anything like that except using the chain, assuming animals in similar chain proximity were more likely to come from similar feedlot lots, right? And then we paid attention to what the lung lesions were, the N being no lesion, L being having a lesion, paying attention to what they were and making sure we were sampling fairly evenly among the lots to make sure that we were trying to make sure that we weren't oversampling a lot that had a lot of no lesions, and we weren't undersampling other lots that were fairly intermediate between the two. So this is an example of something where we could sample a whole bunch of animals. This is an example where we only had a few lung lesion animals and we only sampled a few pairs, okay? Based on that, we basically were able to do something for about $38,000 that would have cost $1.5 million for individual genotyping. Okay, those numbers say something to me. I don't know what they say to everybody else, but I think that's useful. And the nice thing about pooling overall is we could do something similar if we got a feedlot pen or a whole slaughter group, and we could probably tell who the prominent sires in that group were. Okay, which sires had the most influence in that feedlot pen where you have this 30.8 average daily gain or something like that. Okay, and pull that apart and make a genomic relationship matrix type of thing out of the frequencies of animals in that pool. Okay, we could probably do something similar on some of our cow-calf operations if we're able to get a hair sample from all of the animals, combine the hair together and see which sires had the most progeny that year. Okay, it's not just finding the markers that are important for traits, it's finding who's influencing the traits and who's influencing the data set you have, and using that in a weighted approach with traditional BRUP would still bring a whole lot of power to our potential for using these commercial data sets. Research projects will continue, but the grant funding may dry up, at least just for phenotyping, because it's moving on to things that are often more exciting than just the phenotyping alone. Okay, and what do we do next? We'll have less data than the national cattle evaluations, but maybe some of these strategies of using different types of markers will help. Commercial data sources, they all have far more animal numbers per year than we have in our NCE databases. We need to increase the accuracy of recording and genotyping or tying those two genotype sires. We need to provide incentives to commercial operators for data records and try to get situations where that's mutually beneficial. It won't cover all the costs, and we're going to have to find a way to do that somehow if we're going to make progress in these trait complexes. Functional variant methods and DNA pooling could help with some of those